Innate immunity, phagocytic and endocytic barrier. So, immunity being the first immune system that recognizes and gives a response to an external foreign particulate matter, covers or has several layers of defense, of which physical barriers, physiological barriers are the ones that the foreign particulate matters have to cross before phagocytic and endocytic barrier can start acting. So therefore, this barrier actually plays a very important role because many of the cells in this barrier are also able to activate or link the innate immune system with the um, adaptive immune system, which is much more specific and more trained towards a particular foreign specific antigen. So let us look at the learning outcomes of this session. So a very important innate immune defense is ingestion of extracellular particulates by phagocytosis, which is carried out by the effector cells like epithelial cells, macrophages, etc. Phagocytosis comprises engulfing the particulates by pseudopodia. That means the membrane extends into pseudopodia and forms and then engulfs, invaginates, and then engulfs the foreign particulate to form phagosomes, which in turn then fuses with lysosomes to form phagolysosomes. Within the phagolysosomes, the foreign particulates are degraded into smaller fragments by hydrolytic enzymes. The majority of cell components expresses pathogen recognition receptors on their cell surface, and they are able to secrete cytokines and chemokines, thus exhibiting microbicidal mechanisms. So let us understand what are the effector cells. And in fact, the effector cells were first basically discovered or identified by Elie Mechnikov, and who was a Russian zoologist, and he shared the Nobel Prize in 1908 for his studies on phagocytosis. And he linked many people had actually were actually studying this, and they found that there are cells that can take up particulate matter, and therefore they began understanding what is called as endocytosis and pinocytosis. But Ali Mechnikov was actually able to relate it to the importance in terms of it being able to address injury. So if there is a damage or something, then such cells are able to engulf the damaged cells and degrade it. Or they could also give take up the foreign particulates. So therefore, that is how he came to understand that they are associated with giving rise to a defense or giving rise to an immune response. And so he explained, he basically looked at the fact that these effector cells are contributing to what is called as cellular immunity. Paul Ehrlich shared the Nobel Prize, and his study was mainly for antibodies, and that is basically associated with humoral immunity. And we all know today that cellular immunity and humoral immunity go hand in hand. So it's not just one of these which are better than the other. Under any circumstances, when there is a foreign infection, then both cellular and humoral immunity work hand in hand to give rise to a significant immune response and eliminate the foreign particulate matter. Mechnikov recognized the migratory and sessile fixed tissue phagocytic cells in his early studies of invertebrate development, and he did it mainly by microscopy. His studies led to understanding of what is today known as mononuclear phagocyte system. Now, what is mononuclear phagocyte system? So they are a family of cells that comprise bone marrow progenitor cells, which in turn are going to give rise to several different cell types, subcell types that are going to be phagocytic, and of course monocytes and macrophages. So these two we already know are phagocytic. Now, the phagocytic pathway per se is very simple. A phagocyte is able to form what is called as pseudopodia and engulf a particulate matter. Now, interestingly, generally phagocytes are able to recognize particulate matter which are greater than 0.5 microns. And as it forms an early phagosome, there is an NADPH oxidase complex which has been associated with the early phagosome. Now, this early phagosome is able to fuse with several different recycling endosomes present within the system, or they can also be associating directly with the secretory lysosome, and on its surface slowly what is apparently found to be present through Golgi apparatus etc. is the vesicular ATPase, and this vesicular ATPase and a proton pump will gradually make the pH within the phagosome lesser. So you can see that as it combines with the lysosome, its pH decreases, and when the pH comes to a certain level, we all know that the hydrolytic enzymes that are present within the lysosome, sorry, are able to degrade the um, foreign particulate matter. So this is how phagocytosis basically happens, which is similar to endocytosis as a process, and also pinocytosis. Pinocytosis is effectively taking up of liquidish, semi-liquidish matter, and endocytosis is mainly for solid particulate matter. Uh, in many cases, what has been observed is that phagocytosis is also through receptor mediated endocytosis, which means that there are certain receptors that are present on its surface. So the foreign particulate matter would have to bind to the receptor, and then the receptor ligand complex would be endocytosed to form what is called as the phagosome. So these all are basically a methodology of taking up foreign particulate matter and degrading it into smaller pieces. So that is how the particle is eliminated from the circulation. So, interestingly, phagocytosis doesn't just contribute to immunity, that is, innate immune system, but it also contributes to maintaining homeostasis, not just of the immune system, but even other systems. And what has been observed is that it is also able to, or they are also able to engulf damaged cells or injured cells, thereby again maintaining homeostasis. Now, there are phagocytes which are considered to be professional phagocytes. That is basically their primary role is to ingest and degrade the particulate matter. So these professional phagocytes, of course, are derived from or they are originating from hematopoietic stem cells in the bone marrow or from the fetal liver, and then it gives rise to multipotent progenitor cells, which forms lymphoid monocyte progenitor cells. These give rise to a common lymphoid progenitor cells, and these give rise to T cells, B cells, and NK cells. We know that T cells and NK cells are cells that can be phagocytic. Not all T cells are phagocytic, but the cytotoxic T lymphocytes are. NK cells are also phagocytic. Moreover, what has been observed is that when it goes to granulocyte monocyte progenitor, then it forms what is basophil and monocyte progenitor, and that gives rise to basophils, mast cells, eosinophils, and neutrophils. So basically, it is giving rise to granulocytes. So granulocyte monocyte uh, progenitor. And out of all of these all are able to carry out phagocytosis again, especially neutrophils. So, so granulocytic phagocytes are all prevalent in the entry phase of the innate immune response, and each one of them has a slightly different, function, although it will still fall under an innate immune response. So, for example, you have mast cells, considered to be more responsible for allergic reactions because they are able to, more of take into consideration the allergens that are coming into the body, but not restricted to allergens itself. Similarly, eosinophils and basophils can look at parasitic organisms, etc. Neutrophils are generally able to look at all foreign particulate matter. Any type of foreign particulate matter can be addressed by the neutrophils per se, and they are prominent during phagocytosis, so their number increases. So we also know that one of the ways by which doctors are able to predict that there is an infection is by looking at the number of neutrophils, whether they are in the normal range or whether they have gone beyond a certain range. They are also the first to, response to cytokines and chemokines. So the rush to a site where you have more cytokines and chemokines, and that generally happens when the cells that give rise to cytokines and chemokines have been able to encounter a foreign particulate matter on an antigen. Of course, you have the common monocyte progenitor, which can give rise to eventually dendritic cells, monocytes giving rise to macrophages, and also tissue-specific macrophages. And all these mononuclear phagocytes can ingest the foreign particles. So they are professional phagocytes. So you will find that these are what are considered to be professional phagocytes. Then you have what is called as non-professional phagocytes as well. So say for example, these cells' primary role is not phagocytosis, but they do give rise to cytotoxic activity. So eventually, in one way or the other, they are ensuring that the foreign antigen is getting eliminated. So here is an example of natural killer cells. So natural killer cells can distinguish between normal cells and diseased cells. So they can distinguish between healthy cells and the diseased cell. How do they do so? It is because they have receptors which can activate cytotoxicity, and and ligands which can inhibit cytotoxicity. So if the inhibitor receptor is bound to MHC class 1 and the activator receptor is bound to another ligand, then what is understood by the NK cell is that the cell is a healthy cell. So therefore, this degranulation of the NK cell does not take place, or the granzymes etc. are not released, perforins are not released, and therefore the healthy cell is not affected. However, if one of the MHC is one of the things that if not present in a cell, then the NK cell has only the receptor activator binding to the ligand, and because that happens, you have the perforins and the granzymes being released, and that eventually leads to the death of the cell. So this is how it is cytotoxic. Now this has been confirmed by using antibody against MHC1. So if you have an antibody bound to the MHC1, the NK cell will not recognize the MHC1 because the MHC1 is already bound to an antibody. So thereby you have interaction only between the receptor activator and the ligand, and therefore that triggers the NK cell to give rise to all all effector molecules that would eventually lead to uh, harm, or lead to actually a death of the healthy cell. So this is not an infected cell, and yet it is able to do so because of the antibody which is against the anti-MHC. So effectively what we should understand is this, that the NK cell has evolved to identify a healthy cell from a diseased or an infected cell, and that is how it is able to mediate its cytotoxicity activity. It has a natural cytotoxic activity, and that mediation is regulated because of the receptor activator and the receptor inhibitors present on the surface of the NK cells. But you can see over here that NK cells are not phagocytosing. That means they are not engulfing the infected cell and degrading it within it. Their mechanism of action is very different. It is only cytotoxic. So therefore, they fall under non-professional phagocytes. Epithelial and endothelial cells also fall under non-professional phagocytes. So they are definitely responsible for creating a physical barrier. So their first objective would be to prevent the entry of the foreign organism or microbes. They express on their surface pathogen recognition receptors, and these pathogen recognition receptors in turn can recognize certain surface molecules present on the pathogen. So therefore, epithelial and endothelial cells can directly interact with the pathogen, and they can secrete thereby pro-inflammatory cytokines like interleukins and antimicrobial peptides like defensins, cathelicidins, etc. Now let us take the example of alveolar epithelial cells of the lungs. So there are two types of alveolar epithelial cells. Type 1, which comprise 95% of the epithelial cells, and these type 1 express on their surface toll-like receptor 4, which is nothing but a PRR, that is pathogen recognition receptor. And this pathogen recognition receptor is specifically able to recognize and attach to lipopolysaccharides that can be present in bacteria. So this is how you can clearly see how the type 1 alveolar epithelial cells are able to interact with the pathogens. And in response to this interaction, it can produce tumor necrosis factor, it can produce IL-6 and IL-1 beta. All those cytokines and chemokines can be produced by the type 1 alveolar epithelial cells. The other type only comprises 5% of the epithelial cells, but these two are able to produce cytokines and chemokines, and both the type 1 and the type 2 alveolar epithelial cells are also able to produce surfactant proteins like surfactant protein 1 and 2. And these surfactant protein 1 and 2 are in turn able to initiate processes of chemotaxis and enhanced phagocytosis. So you can see how indirectly it is recruiting leukocytes and it is mediating pro-inflammatory events. So this is again very important. They themselves are not engulfing, but they are calling cells that can engulf the pathogens. Platelets are also having an action or having a positive action on innate immune response through phagocytic barrier. They are able to control bleeding. Now, in the formation of clot, what can therefore happen is that the mesh that is formed because of the clot can also hold within it several pathogens. So therefore, it is able to create a kind of defensive mechanism of not allowing the pathogens to move farther from the clot. They can express pathogen recognition receptors again, and this enables it to interact with the pathogens directly. They also have surface adhesion molecules such as P-selectin. So, because of the interaction of the pathogen recognition receptors with the pathogen, these platelets are able to produce cytokines and chemotactic molecules, which in turn will recruit leukocytes. The surface adhesion molecules being present, such as P-selectin being present, they are able to bind to several adhesion molecules on the endothelial layer and therefore lead to what is called as extravasation, and that local extravasation will lead to inflammation at the region where the antigen is present. So it helps in interacting with leukocytes and endothelial cells, and both of these are able to mediate pro-inflammatory events. So let us make the conclusions. The mononuclear phagocyte system originating from bone marrow progenitors comprises professional phagocytes such as macrophages, monocytes, neutrophils, etc. The phagocytes are able to ingest the foreign particulate and with the help of hydrolytic enzymes of the lysosomes degrade these. Non-professional phagocytes, and in this case also many of them have the mechanism of oxygen dependent lysis as well, oxygen dependent degradation as well as a non-oxygen dependent mechanism. Non-professional phagocytes such as NK cells, epithelial, endothelial cells, etc. are not directly phagocytic, but they can mediate phagocytosis, and NK cells themselves are cytotoxic. Another note over here is that dendritic cells also can take up antigens, and they can act as antigen presenting cells along with macrophages. So both professional and non-professional phagocytes recognize the exogenous particulates due to their ability to express pathogen recognition receptors. The innate immune system has layered its defense with facilitated endocytosis and phagocytosis by cells, that being one of the layers of non-specific defense, but heightened immunologic responses with some cells also aiding in forming what is called as adaptive immunity. So some of these cells are links between innate immune system and the adaptive immune system. Thank you.